Thank you very much, Rebecca, for the invitation. I guess I'm supposed to have some kind of microphone here, right?
No, you need.
No, I don't need it. Okay, very good. So you know, Rebecca has been at the very top of our research field for over 20 years, and she's been at EML and HITS for you know, maybe 10 or 15, I'm not sure. But what I can say now is that HITS has expanded the number of research groups it has, and it's added several really top-level groups. So HITS has arrived now, yeah? HITS has arrived. HITS is really world-class. So the way I see it, as far as HITS is concerned, is you know, it's just beginning. And the impact that HITS will have worldwide is going to be very interesting to see in the next decades. So that's HITS. KTS has been active for much longer, of course. But I'm interested in what this thing means here. What is it? This is this extra dot on the logo. What could it be? Is it some a galaxy that doesn't have quite enough mass in the simulation? Or is it in the cluster that HITS has, one of the nodes that you have to take out because it doesn't function anymore? Or maybe these are the group leaders in HITS, you see, and this is a crazy group leader who's a little bit different. And I would like to suggest that that's Frauke Greta. I'll give it an argument as to why I think it might be Frauke Greta later on. So, we're going to talk about saving the environment, curing disease, and I missed one of them out. Endless source of renewable energy. How about that? We'll add that in as well. So about 10 minutes to solve each of these three problems. Is that a reasonable amount of time? So we'll start off with the disease. Oops, I've got to do that here. That's right. And actually 10 days ago, both myself and my wife were hit by this, yeah? 10 days ago, we were hit by the flu. Now, my wife, she took a computer-designed pill, and she was better in 1 day. 1 day, she took a pill that was designed by computer. Now, I didn't bother to take the pill, and so I'm still suffering from the damn thing. And so if I cough halfway through, you'll understand why. I didn't bother to take the pill. So, how was this designed? Now, this next slide is a very elementary biology slide. Very elementary biology. Now, in America, you have to put these slides up because you know, when one talks to politicians and businessmen, etc., they don't know the elementary biology. So I presume all of you know this, but I'll go through it anyway here. We talk about genes. Genes are sequencers of chemical letters in DNA. So, you know, gene is a sequence of chemical letters, and that's then translated by a machinery into a sequence of chemical letters in a protein. That's what a gene does. It translates a sequence of chemical letters in DNA, and it gets translated into a protein which folds up into a wonderful, exquisite three-dimensional architecture. That's the protein structure. And these proteins do the various functions of life. Some of them are antibodies, for example. Others are enzymes that make the chemical reactions that the cell needs work quickly. Others are hormones. Others are mechanical proteins and things like that. And they do all the work of life. And what became clear, I'm going to go back again. What became clear in the 1970s and before was that the technique of X-ray crystallography could solve the structures of these proteins. We haven't yet managed to fold these proteins up ab initio in a computer. We don't know how to do that yet. But using experimental X-ray crystallography, some of these structures were beginning to be solved. So immediately when the first structures came out, people thought, well, you know, this is what nature looks like. If we want to design a drug, we should make use of this three-dimensional structure. And so let me see, I have to figure out how to make this movie go now. I think this is it. And here. Computers were used then to dock these small molecule trial chemicals into proteins such as this one. This is a cancer target protein. And to do calculations to try and figure out which chemicals might fit into the binding site of the protein target that one is interested in. And the people at HITS may not be aware of it, but there's one of their group leaders that was in right at the beginning of this. There she is. That's Rebecca again. In the late 1980s and early 1990s, she helped develop one of the first computer programs, it might even have been the first, that tried to calculate whether a chemical would bind in a given pocket that a protein target might have. And she did that at Oxford with Peter Goodford, and the program was called, as I guess many computer programs are called, GRID. It's the GRID program for drug design. And she was a major developer of that in the 1980s and 1990s. Now, the result of that program was that people took it and used it and designed these things. Relenza and Tamiflu. You've heard of those probably, or you've probably heard of Tamiflu at least. No? Relenza was the first one. They took the three-dimensional structure with the GRID program that Rebecca helped develop, and it was fairly easy for them to fit that into the three-dimensional structure and then pretty quickly come up with a drug that worked against the flu, and then finally another one, Tamiflu, that's even easier to take. And that is the computer-designed pill that my wife took last week and that I forgot to take and I'm still suffering from. So that's called structure-based drug design. You design drugs based using computers and experimental information on structures of proteins. And of course, when it works, it's very useful. Now, what about this dot over here? That's Frauke Greta. Here she is. When she was a diploma student in my lab in Heidelberg, she did work that resulted in a paper that was rather different from the methods that had been used before, because Frauke's paper showed that you can actually use first principles to design drugs. That's to say, quantum chemistry. You may all know that quantum mechanics is the basis of how molecules interact, but nobody had used that directly. Frauke did that, so it's rather off on a limb already. And since then, it's created quite a stir, and a number of other groups have done that. That's not the only reason that she's out there in the dot, though. After she'd done that, this is around about 10, 12 years ago, I guess, she came up to me with a crazy idea, at least what I thought was a crazy idea. She said, can I do a PhD in China? PhD in China. Can I do a PhD in China? Now, this is 10, 15 years ago, remember? And I said, no, you cannot do a PhD in China, because I didn't think that the Chinese science was sufficiently developed at that time. Yeah? They hadn't yet. But she was a naughty girl, and she went to China anyway, and she did a great PhD there. She went to Shanghai. This is Shanghai, you see. And she did a great PhD there and helped set up a Max Planck Institute, and then she came back. Now she's a professor here and the group leader at HITS, right? So she proved me wrong. So that's one reason why she's out there. Another reason is that the subject that Frauke has been doing is another crazy subject, and it's to calculate forces in protein molecules. All the rest of us thought this was impossible to do because the forces fluctuate so much in simulations that you'd never get a sensible answer. But she showed that if you do the simulations long enough, you can get a sensible answer out of it, and it can provide lots of information of use when you want to understand how a protein molecule moves. So that's why she's that weird dot out there, I reckon. So we're now with structure-based drug design. Well, you see, in the United States, the researchers there are very modest. And so we have this supercomputer at Oakridge National Lab that we've modestly called Titan. Yeah? Now, Titan, we think, is the world's most powerful supercomputer. It's faster than anything else. Actually, on the official list of supercomputers, it's at number 2. And there's one in China that's more powerful. But you see, we're not sure about that because we think the Chinese just put it together to do the test and then took it apart again, right? That's what we think. But anyway, it's number 2 in the world right now. And maybe you know the truth behind this. We can't find the truth. So with this computer, you see, what you can do is you can take a library of millions of chemicals, and when medical people or biomedical scientists have identified a target, you can see using the supercomputer by doing lots of calculations in parallel which one is most likely to bind, using methods similar to the ones that Rebecca developed 20 years ago. That's virtual high throughput screening of chemicals using supercomputers. Now to do this though takes a bit of work. What you want is if you have a computer cores, our supercomputer is like having 2 million laptops piled up on top of each other, and you want them all working. You don't want some of them not to work while others don't. So when we started doing this docking in 2008, this would be the number of cores you use, and this would be the speed up of the time to do the calculation. And this is the ideal case, yeah? When you double the number of cores, you want to go twice as fast. So what was happening is things were leveling off at around about 100 cores. Then our computer scientists got onto it, and now, a couple of years ago, we managed to scale up to about 100,000 cores, which allowed us to screen, do this high throughput screening of compounds. You do 500,000 in about 15 minutes. And now we are scaling these things up to 1 million cores on the Titan supercomputer. So we can do it all very quickly for drug companies and people like that give us money. But doing that sort of brute force stuff is interesting, but what many of us, including Rebecca and Frauke, have realized is that protein molecules are not static. This is a kinase, it's another cancer target, and you can see that if you do a simulation of how the atoms move in a protein, the active site, which is here, that's the site that you want to block, is really quite flexible, and it changes shape quite a lot. Rebecca's working on getting these shapes calculated now, but one thing you can do is you can take snapshots of this simulation and use each individual snapshot as a target to find a molecule to fit in, yeah? So that's what you can do when you get lots of computer power. I want to show you one result of our work concerning thrombosis now, which used that. Oops, there we go. This is blood clotting. Now what you want to do, what doctors tell us, is that they sometimes want to make people's blood thicker, and sometimes they want to make it thinner. And they would like small molecules. These are drugs that could do either of those. We were told that, well, biologists tell us there are these cascades of interactions between proteins that form, and then if we hit one of them in particular, interactions between proteins, we should be able to modulate how thick somebody's blood is. So this was done using this dynamical simulation that I showed you in the previous slide, and without going into the details, 10 molecules resulted from that would modulate coagulation in both ways, up and down, make it coagulate more or less, which is just what the pharma company wanted that worked with us, and that 8 of those 10 would not have been found if we just used the crystal structure. What do I mean by that? Just the static structure that the experimental X-ray crystallographers give us, thinking that's a key to bind the lock-in, would not have found 8 of those 10 structures. You need the dynamically modulating shapes of the binding site to do that. So what about the future? We're talking about thinking beyond the limits here in this symposium. So let's first of all look at one of the problems in drug design. The problem is that it doesn't work very well. Drug development, typically before you get a drug approved by, in the USA, the FDA, you've gone through about $1 to $1.5 billion of expense. You've screened experimentally now maybe 1,000 compounds, and these become HITS, pharmaceutical HITS this time, and then they get developed and modulated, and you then go into clinical trials, which are very expensive, before you get one of them that's approved. So why do all these candidates fail? Well, two reasons. One is efficacy, meaning they don't do the biological thing that you want them to do. For example, they don't clot blood or whatever, or thin blood. And the other is safety, which means they do something you don't want them to do. It means they're toxic. Think of all the cancer chemotherapies that have horrible side effects that we don't like. So what do we need to do? Well, I think for the future, we need computationally to begin right here, day one of the drug design process. And we need to develop methods that can predict which of these compounds are going to fail due to safety or efficacy. That's the challenge. And the fact is that even if you can do 10% better than random, you are going to save years. This is a 10-year-long process. You're going to save years and hundreds of millions of dollars in a drug design process. So you can just do a little bit better than random in helping weed out those drug candidates that are not going to be safe or not going to be efficacious. So how do we do that then with structures? I think in the future, what's going to happen is there'll be a large amount of development on predicting side effects. So if you have a chemical, what was done with Tamiflu, for example, was people found a molecule that would bind into a certain protein on the flu virus called neuraminidase. So they found that. But what you can do now with all this computer power we have is to see if that same molecule will bind to other proteins that you don't want it to bind to, by again doing the docking calculations. And that way you can begin to calculate side effects of possible chemicals. So we're beginning to do that now, beginning to have some success, but I think having that right at the beginning of the drug design process will be very important. And the second thing goes back to all those genes that we heard about from Rebecca. What's going to happen a little bit later in the future, when we talk about personalized medicine, medicine that works for individual people, is, well, maybe all our children will all have their genomes sequenced. There are hundreds of thousands of genomes being sequenced now in the United States, at least. So they've all got their personal gene sequences. Structural biology and computational biology then will be advanced enough that we'll all be able to calculate our individual protein structures that are a result of our individual gene sequences. So we'll all have somewhere on a computer disk, or whatever there is then, our own structures of proteins that every individual has, and we'll be able to dock millions of molecules into each of these to see which ones work for individual people, right? So it's individual structure-based drug selection, if you like, in personalized medicine. That's something to aim for the future. It's not here right now. Okay, so I'll switch briefly to energy, to something that's happening in the hills of Tennessee, the Appalachian Hills of Tennessee. This is comes a little later. What we're talking about is a way of producing energy that doesn't produce greenhouse gases and that's very, very efficient, and that would be second generation biofuel production, that's cellulosic ethanol. Now with that, you start off with something the experts call cellulosic biomass, but I call plants. It's the same thing. It's biological stuff that's got cellulose in. The next step is you want to hydrolyze the cellulose. That breaks it down into sugars. And then you ferment the sugars into ethanol. Now, up in the hills of East Tennessee, I don't know if anybody knows East Tennessee, it's got the Smoky Mountains in, and it's got it's not like the Odenwald, yeah? In the Odenwald, you've got very sort of, ordentlich villages with cuckoo clocks. Oh, that's the Schwarzwald, but the Everything's very ordered. You know, in the wilds of East Tennessee, police cars do not dare drive up there. Police cars don't go up there. And one of the main reasons is this, is they know how to ferment. They know how to ferment. This is the hills of East Tennessee. You may recognize this as an illegal moonshine still. They're all over the place. You don't go up there with those. And this guy, his name is Popcorn Sutton, and he's one of the main moonshine producers in East Tennessee. You know, it's a big business, I'll tell you that, a big business. And so anyway, fermentation in Tennessee, we know how to do that. That's pretty easy, the fermentation. But the difficult bit is the hydrolysis. And the word we use in our Bioenergy Science Center is that biomass is recalcitrant to hydrolysis. Like Frauke was recalcitrant when I told her to stay in Heidelberg, not go to China. Yeah? So how do we overcome this recalcitrance? Well, it's all to do with molecules interacting again. The main way of producing this ethanol is through these wonderful, shining enzymes. They are called cellulases, and they chew up cellulose strands into glucose. These are little glucose things, yeah? But the problem is the cellulose strands are all packed together and difficult to get at. They're physically difficult to reach. And there's this brown stuff that's in the way. Brown stuff in the way. That's called lignin. And that's the other major component of plant cell walls. So we need to find a way of getting access to the cellulose for the cellulase enzymes. So how do we do that? Well, we use big supercomputers like this one. The simulations, I don't know if this is going to actually click. Nope. I don't seem to be able to run the simulation because the arrow's in the way or something. Anyway, if I could, try once more to click this without clicking the arrow. Anyway, there we go. That's going to do it, isn't it? No.
Just click the image.
What did you say?
Click the image.
I didn't hear. Oh, it's going to... No. Anyway, you would see the lignin sticking to the cellulose. It would gradually, during this molecular dynamics simulation, come. Yeah, you go on. Here's another spot on the outside, I'll tell you. What were you saying?
Click right on the middle. Right in the middle of the image.
Middle of the image. All right, I'll try that then. Where are we? There we go. Click right on the middle of the image. Oh, yes, you are clever. You see? I hate computers. Don't you all hate computers here? I hate them. I really do. I hate the things. Can't even switch them on. But anyway, you see the lignin, which is the green stuff, is sticking to the cellulose. And this multi-million atom supercomputer simulation using the Titan machine at Oakridge then allows us to identify the physical and thermodynamic contributions to what makes the lignin stick. Now, we don't want the lignin to stick. And so we try different solvents out, and we found one now. It's a kind of secret, but I'll tell you what it is. It's called THF. And this THF solvent will 100% dissolve up biomass and completely overcome its recalcitrance to hydrolysis. So that works. Simulations help there. Now, you can do ever larger simulations. If I click on this, do you think it'll work? Yes, you see, this is a 24 million atom simulation now that's taking hundreds of thousands of processes, and it's not only of the cellulose, which is now annoyingly brown, it's also the lignin, which is now blue, and the cellulase enzymes, yeah? So you have the enzymes trying to get past the lignin, and this kind of simulation model we're using now to develop methods to overcome the recalcitrance to hydrolysis of biomass. So we're getting ever larger, you see. We need these machines. What about when we get to the exascale? Now, some of you will know what the exascale is. It's a computer that's capable of doing, I think, 1 million floating point operations per second. Yeah? An exaflop of computing power. And we'll be there in a few years. The problem is with that is it takes the power of a small town just to run the computer. So we have a power problem. When we get to that, though, in principle, we'll be able to simulate, using these dynamical methods, molecular dynamics, 10 to the 10 atoms, a system of 10 to the 10 atoms, which is all the atoms in a living cell. So a complete living cell at atomic detail, in principle. We'll have enough computer power to do that. In practice, it'll be very difficult to know where to start from to do that. But it gives an idea of the kind of thing we'll be able to do. So for the last few minutes of the talk, then, I want to keep talking about energy. This energy here. This is the Castle Bravo nuclear test at the height of the Cold War in 1954 that was made with nuclear material from Oakridge National Lab. And when they made this test, they had to produce the hydrogen bombs, they had to separate lithium isotopes out, and they used lots of mercury to do that. They dumped the mercury at the dumping sheds, you see here. And unfortunately, it got into the ground and polluted the ground, and that's a problem. Now, this was done at what's now called the Y-12 Nuclear Weapons Institute. It's next door to our institute. And that's where most of the nuclear material for bombs is made in the USA. And it's a fortress, Y-12. You don't get in there very easily. It's very, very difficult to get in. It's very well protected, except an 84-year-old nun, the name of Sister Megan Rice, she managed to break into it 2 years ago. So she clipped, with two accomplices who are in their 60s, she clipped through the fence, and the alarm went off, and the guards thought it was just another deer, because there's lots of deer all around. She got in, she went to the inner sanctum of this nuclear weapons establishment, and stayed there for 3 hours before her and her friends were found. As a result, they fired all of the security personnel at this institute, and they had to replace them with somebody else. That's an aside. Getting back to mercury, the mercury that went into the ground then has polluted Oakridge, and this is a sculpture based on liquid mercury in Chicago. It then gets into the streams, the rivers, etc., and it's a real pain. And what happens is that bacteria in the streams methylate it. And they create methyl mercury, which is even worse. That's a real toxin. And now, when the bacteria create methyl mercury, tiny organisms that you can't see will eat the bacteria, and then bigger ones eat those, and small fish eat those, and big fish eat those. And every time something eats something, the methyl mercury gets more and more concentrated. So if you eat an old fish, like albacore tuna or swordfish, it's going to have a high concentration of methyl mercury in. Now this guy, he was the CEO, still is, of the Imax Corporation. That's this 3D movie corporation, right? And he was a health freak, and he used to go jogging every day, enjoyed that, and he loved tuna sushi. Yeah, loved tuna sushi, so after a while, a few years ago, he found that he had to hold on to lampposts when he was jogging. And then a few months after that, he could not walk anymore. And that was due to methyl mercury poisoning that was found out very cleverly. So he stopped eating tuna, and he's sort of 60% recovered now. Others haven't had quite that luck. There was the Minamata May disaster in Japan that killed 2,000 people, 10,000 deaths in Iraq through methyl mercury containing fertilizer, and still the odd case of this poisoning now. So at the end of this, there was one question that environmentalists wanted to know. How do bacteria methylate mercury? They are the culprits. They are the bad microorganisms that turn relatively inert elemental mercury into methyl mercury. And we've known since the Minamata Bay disaster in Japan, which is in the 1960s, we've known that it's bacteria that do this, but when you do these phylogenetic trees like we just saw, it never told anybody anything. There was never any relationship between those bacteria that will methylate mercury and those bacteria that don't. And looking at the gene sequences didn't help either. So 40 years went by, nobody knew how bacteria do this, and we need to know that, yeah? So Jerry Parks, what he did in our lab, I'm not going to go through this, he thought about chemistry. He said the chemistry that has to happen is carbanion transfer to the mercury 2+. So that was different. And then he figured out what you'd need for that. What you need is vitamin B12. So bacteria need their vitamins as well, not just us. And these bacteria that have vitamin B12, he thought maybe those are the ones that can do this chemical reaction to methylate mercury. So he suggested that to the experimentalists. Before he did that, he found in the bacteria a certain gene that he called HGCA that contains the chemical groups that can bind vitamin B12. And built a model of it showing that the chemistry could take place in principle that would methylate the mercury. He found another gene that would supply the electrons needed, and then we went to the experimentalist and he said, well, there we are, there are your genes, test them. Now, experimentalists are a pain sometimes because they don't do what you tell them to do, right? Computational people, we make predictions, and experimentalists say, so what, we're not interested. But eventually they did test that, and it turned out that it was the correct prediction. They did the tests that you do by knocking out one of the genes, knocking out the other one, knocking out both of them. Methylmercury wasn't produced. That was about a year ago, whereas if you have the genes, it does produce methylmercury. And now there's about 50 research groups worldwide that are using this as a tool for figuring out where methylmercury is produced in the environment. So the message is, and this is the last slide, that the computational science, which here was in finding this protein in a data bank, building a structure, and showing computationally that the chemistry could take place that was needed. That's the computational science. What it did was to unify different research fields. In this case, the approaches that were used were chemical reasoning, three-dimensional thinking. The computational part was structural bioinformatics, comparing structures of molecules and figuring out what function they would have. With experimental genetics and microbiology, you had to get all these teams, in fact 3 teams together, to do that, to solve this problem. And I think for the future of computational science, the future is going to be in unifying different types of experiment. Experimentalists tend to be rather narrow-minded people that only can produce one measurement for you, but there are different types of experimentalists that produce different types of data, and integrating that into physical models, whether it be of the universe, whether it be of the living cell, whether it be in this case of the cycling of toxins in the environment. This kind of unified model is something that science is moving towards. In the USA, all of the major funding agencies, the governmental ones, are emphasizing computational science now, and they're emphasizing its ability to unify experiments across time and length scales. And I think that will be the future. I think HITS has a very rosy future in helping in this integration. With that, I'll thank you for your attention.